Hi class, this is your instructor, Skylar Huff. We're now in Chapter 4. This is cell structure. All organisms are composed of cells. The gossamer wing of a butterfly is a thin sheet of cells, and so is the glistening outer layer of your eyes. The burger, or the tomato you eat, is composed of cells, and its contents soon become part of your cells. Some organisms consist of a single cell, too small to see with the unaided naked eye. Others, such as humans, are composed of many specialized cells, such as fibroblasts, which is the cell shown in the striking fluorescence micrograph you see here to the right. Cells are so much a part of life that we cannot imagine an organism that is not cellular in nature. So here we'll take a closer look at the structure of the internal of cells. Throughout the chapter, we'll go from cell theory to prokaryotic and eukaryotic cells. We'll go throughout the endomembrane system. We'll get to mitochondria and chloroplasts, the cytoskeleton briefly, as well as briefly with extracellular structures and cell movement, and end things with cell-to-cell interactions. Let us begin. Cells are too small for you to be able to see. Although there are exceptions, a typical eukaryotic cell is 10 to 100 micrometers in diameter, and most basic prokaryotic cells are only 1 to 10 micrometers in diameter. Because cells are so small, they were not discovered until the invention of the microscope in the 17th century by the English natural philosopher Robert Hooke, who was the first to observe cells in 1665, naming the shapes he saw in cork cellulae, or small rooms. They are known as cells to us. Another early microscopist, Dutch Antoine van Leeuwenhoek, first observed living cells when he termed animalcules, or little animals. After these early efforts, a century and a half passed before biologists fully recognized the importance of cells. In 1838, German botanist Matthias Schleiden stated that all plants are aggregates of fully individualized, independent, separate beings, namely the cell themselves. And in 1839, German physiologist Theodor Schwann reported that all animal tissues also consist of individual cells. Thus, the cell theory was born. It was proposed to explain the observation that all organisms are composed of cells. It sounds simple, but is a far-reaching statement about the organization of life. In its modern form, this cell theory includes the following three principles. All living organisms are composed of one or more cells, and the life processes of metabolism and heredity occur within these cells. Secondly, cells are the smallest living things, the basic units of organization of all organisms. Thirdly, cells arise only by division of a previously existing cell. Although life likely evolved spontaneously in the environment of early Earth, biologists have concluded that no additional cells are originating spontaneously at present. Rather, life on Earth represents a continuous line of descent from those early cells. Most cells are relatively small for reasons that relate to the diffusion of substances into and out of them. The rate of diffusion is affected by a number of variables, including surface area available for diffusion, temperature, concentration gradient of diffusing substance, and the distance over which the diffusion must occur. As the cell size increases, the length of time for diffusion from the outside membrane to the interior of the cell increases as well. Larger cells need to synthesize more macromolecules. have correspondingly higher energy requirements, and produce a greater quantity of waste. Molecules used for energy in biosynthesis must be transported through the membrane. Any metabolic waste must be removed, also passing through the membrane. The rate at which this process occurs depends both on the distance to the membrane and the area of membrane available. For this reason, an organism is made up of many relatively small cells, has an advantage over one composed of fewer larger cells. The advantage of small cell size is readily apparent in terms of the surface to area volume ratio. As a cell's size increases, its volume increases much more rapidly than its surface area. For a spherical cell, the surface area is proportional to the area of the radius, whereas is proportional to the square of the radius, whereas the volume is proportional to the cube of the radius. Thus, the radii of two cells differ by a factor of 10. Thus, if the radii of two different cells vary by a factor of 10, the larger cell will have 10 to the second, or 100 times the surface area, but 10 to the third, or 1,000 times the volume of the smaller cell. The cell's surface provides the only opportunity for interaction with the environment, because all substances enter and exit a cell via this surface. The membrane surrounding the cell plays a key role in controlling the cell function. Because small cells have more surface area per unit of volume than large ones, control over cell contents is more effective when cells are relatively small. Although most cells are small, some quite large cells do exist. These cells have apparently overcome the surface area to volume problem by one or more adaptive mechanisms. For some cells, such as skeletal muscle cells, they have more than one nucleus, allowing the genetic information to be spread around a large cell. Some other large cells, such as neurons, are long and skinny, so that at any given point within the cell is close to the plasma membrane. This permits diffusion between the inside and out of the side of the cell to still be rapid. So there is figure 4.1 showing the surface area to volume ratio. As a cell gets larger, its volume increases at a faster rate than does its surface area. Microscopes. Other than egg cells, not many cells are visible to the naked eye. Most are less than 50 micrometers in diameter, far smaller than the period at the end of this sentence. Look at that period. So to visualize cells, we need the aid of technology. The development of microscopes and their refinement over centuries has allowed us to continually explore cells in greater detail. How do we study cells if they are too small to see? The key is to understand why we can't see them. The reason we can't see such small objects is limited to the resolution of the human eye. Resolution is the minimum distance two points can be apart and still be distinguished as two separate points. When objects are closer together, about 100 micrometers, the light reflected from each strikes the same photoreceptor at the rear of the eye. Only when objects are farther away than 100 micrometers apart can the light from each strike different cells, allowing your eye to resolve them as two distinct objects rather than one. Types of microscopes include. So one way we overcome the limitations of our eyes is to increase the magnification so that small objects appear larger. The first microscopists use a glass lens to magnify small cells and cause them to appear larger than the 100 micrometer limit that is imposed by your and my eye. The glass lens increases focusing power. Because the glass lens makes the object appear closer, the image on the back of the eye is bigger than it would be without the lens. The modern light microscope now. They operate with visible light using two magnifying lenses and a variety of correcting lenses to achieve very high magnification and clarity. The first lens focuses the image of the object on the second lens which magnifies it again and focuses it on the back of the eye. Microscopes that magnify in stages using several lenses are called compound microscopes. They can resolve structures that are separated by at least 200 nanometers. Light microscopes, even compound ones, are not powerful enough to resolve many of the structures within cells. For example, a cell membrane is only 5 nanometers thick. Why not just add another magnifying stage to the microscope to increase its resolving power? This doesn't work because when two objects are closer together than a few hundred nanometers, the light beams reflecting from the two images start to overlap each other. The only way two light beams can get closer together and still be resolved as if their wavelengths are shorter. So one way to avoid overlap is by using a beam of electrons rather than a beam of light. Electrons have a much shorter wavelength, and an electron microscope employing electron beams has 1,000 times the resolving power of a light microscope. Transmission electron microscopes, so called because the electrons are transmitted through a specimen to visualize it, can resolve objects only 2 nanometers apart, which is only twice the diameter of a hydrogen atom. A second kind of electron microscope, the scanning electron microscope, beams electrons onto the surface of the specimen. The electrons reflected back from the surface together with other electrons that specimen itself emits as a result of the bombardment are amplified and transmitted to a screen where the image can be viewed and photographed. Scanning electron microscopy yields striking 3D images. This technique has improved our understanding of many biological and physical phenomena. Although resolution remains a physical limit, we can improve the images we see by altering the sample. Certain chemical stains increase the contrast between different cellular components. Structures within the cell absorb or exclude the stain differentially, producing contrast that aids resolution. So there we have size scale of cell components shown in figure 4.2. All cells share simple structural features. The general plan of cell organization varies between different organisms, but despite these modifications, all cells resemble one another in certain fundamental ways. Before we begin a detailed explanation of the cell structure, let's first summarize 4 features all cells have in common. A nucleoid or nucleus where genetic material is located, cytoplasm, ribosomes to synthesize proteins, and a plasma membrane. Centrally located genetic material. Every cell contains DNA for genetic material, and in prokaryotes, the simplest organisms, this consists primarily of a single circular molecule of DNA. It is typically found near the center of the cell in an area called the nucleoid. This region is not surrounded by a membrane. By contrast, eukaryotic cells are more complex, with DNA organized into linear chromosomes segregated into a nucleus, organized into linear chromosomes in that nucleus, which is surrounded by a double membrane structure called the nuclear envelope. In all cells, the DNA contains the genes that code for proteins synthesized by the cell. A semi-fluid matrix called the cytoplasm fills the interior of the cell. The cytoplasm contains all of the sugars, amino acids, and proteins the cell uses to carry out its everyday activities. Although it is an aqueous medium, cytoplasm is more like jello than water. And that is due to its high concentration of proteins and other macromolecules. The fluid part of the cytoplasm, which contains organic molecules and ions in solution, is called the cytosol. Membrane-bound organelles are usually specialized for a particular function and contains specific proteins, creates compartments in the cytoplasm. Ribosomes are large macromolecular machines composed of RNA and protein that synthesize all cellular proteins. Ribosomes use information from the genome and other accessory molecules to direct the synthesis of proteins by sequential addition of amino acids. The plasma membrane encloses the cell and separates its contents from its surroundings. The plasma membrane is a phospholipid bilayer about 5 to 10 nanometers thick, with proteins embedded in it. Viewed in a cross-section with an electron microscope, membranes appear to be two dark lines separated by a lighter area. This arises from tail-to-tail organization of phospholipid molecules that make up the membrane. The proteins of the plasma membrane are generally responsible for a cell's ability to interact with the environment. Membrane proteins give cells identity and provide for a variety of functions including transport and communication with other cells and the environment. The role of the plasma membrane is so important we will study it in detail in our next chapter. Prokaryotes are very important in the ecology of living systems. Some harvest light by photosynthesis, others break down dead organisms and recycle their components. Still, others cause disease and are used in many industrial processes. Prokaryotes have two main domains, archaea and bacteria, introduced here and discussed later on at the end of this course in Chapter 27. When cells were visualized with microscopes, two basic cellular architectures were recognized, eukaryotic and prokaryotic. These terms refer to the presence or absence, respectively, of membrane-bounded nucleus that contains genetic material. In addition to lacking a nucleus, prokaryotic cells do not have an internal membrane system or numerous membrane-bound organelles. Our view of prokaryotic cell structure has changed significantly over the years. They have been described as small, simple cells consisting of a plasma membrane containing cytoplasm with genetic material and ribosomes, and encased by a rigid cell wall. While accurate as far as it goes, this was an oversimplification. Prokaryotes were thought to lack any membrane-bound organelles, which form compartments in eukaryotes. While still accurate to say that there are no organelles common to all prokaryotes, particular species do have specific organelles that provide specialized functions. The best studies of these is the magnetosome, found in bacteria that can move along a magnetic field. Magnetosomes consist of spherical membranes containing iron oxide crystals. These are arrayed in a chain by an attachment to an external fiber, which allows the cell to align with the magnetic field. Both bacterial and archaeal species may also have internal membrane-bound storage structures. Prokaryotes can also have enfoldings of the plasma membrane that serve to segregate metabolic reactions. Some bacteria also contain cellular compartments bounded by a semipermeable protein shell. These bacterial microcompartments range in size from 40 to 400 nanometers and act to isolate specific metabolic processes or to share particular substance. While the structure is quite different, these compartments are functional analogs of eukaryotic organelles. Prokaryotes were also thought to lack an elaborate cytoskeleton found in eukaryotes, but we have now found that they have molecules related to both actin and tubulin, which form two of the cytoskeleton elements described later in this chapter. The strength and shape of the cell is determined by the cell wall and not these cytoskeleton elements. However, cell wall structure is influenced by cytoskeleton. For instance, the presence of an actin-like fiber running the length of the cell leads to a perpendicular cell wall fibers that produce a rod-shaped cell. This can be when this protein is removed. Cells become spherical rather than rod-shaped. During cell division, cell wall deposition is influenced by tubulin-like FTSZ protein. Most bacterial cells are encased by a strong cell wall, composed of peptidoglycan, which consists of a carbohydrate matrix cross-linked with a short polypeptide units. Cell walls protect the cell, maintain its shape, and prevent excess excessive uptake, or loss of water. The exception is the class Molecules, which includes the common genus Mycoplasma, which lacks a cell wall. Plants, fungi, and most protists have cell walls, but with a chemical structure different from that of peptidoglycan. Antibiotic drugs used to combat bacterial infections often target their cell walls. For example, penicillin and vancomycin interfere with the ability of bacteria to cross-link the peptides in their peptidoglycan cell wall. Like removing all the nails from a wooden house, this destroys the integrity of the structural matrix, which can no longer prevent water from rushing in and swelling the cell to bursting. Some bacteria also secrete a jelly-like protective capsule of polysaccharide around the cell. Many disease-causing bacteria have such a capsule, which enables them to adhere to teeth, skin, and food, or practically any surface that can support their growth. We have much to learn about the physiology and structure of archaea. Many archaeans are difficult to culture in the lab, as shown recently by the decade-long effort required to culture a single deep-sea archaean. It is much easier to identify archaeal genomes in environmental samples. So we know more about archaeal genomics than archaeal physiology. The cell walls of archaea are more diverse than bacteria, containing polysaccharides and proteins, and possibly even inorganic components. One feature that distinguishes archaea from bacteria is the structure of their membrane lipids. Archaeal membrane lipids include saturated hydrocarbons that are covalently attached to the glycerol at both ends, which forms a monolayer membrane. These features seem to confer greater thermal stability to archaeal membranes, although the trade-off seems to be an inability to alter the degree of saturation of the hydrocarbons, limiting their response to changing environmental temperatures. So the cellular machinery that replicates DNA and synthesizes proteins in archaea is more closely related to eukaryotic systems than to bacterial systems. Even though they share a similar overall cellular architecture with bacteria, archaea seem to be more closely related on a molecular basis to eukaryotes. Bacteria, archaeal, and eukaryotic cells have external structures for motility. Long filaments made of proteins that are superficially appear similar, but actually are three evolutionary distinct structures. Bacterial flagella consist of protein rings embedded in the plasma membrane and a cell wall with long protein fibers extending from this. The analogous archaeal structure, called the archaeolum, consists of a structure that appears to be related to different bacterial external structure known as a pilus. This is formed by a disk of membrane proteins, which protein filaments extending from the cell. Both bacterial and archaeal structures rotate like a propeller, while eukaryotes have a whip-like flagellum. However, the motive for force and rotation is different. Bacteria use the proton pump, similar to that used in eukaryotic mitochondria, while archaea use the hydrolysis of ATP. The independent evolution of the different structures with similar function is called convergent evolution. Eukaryotic cells are far more complex than prokaryotic cells. The hallmark of the eukaryotic cell is compartmentalization. This is achieved through a combination of extensive endomembrane system that weaves through the cell interior by numerous organelles. These organelle membrane-bounded structures that form compartments within which multiple biogeochemical processes can proceed simultaneously and independently. Plant cells often have large membrane-bound sacs called a central vacuole, which stores proteins, pigments, and waste materials. Both plants and animals contain vesicles. So vesicles are smaller sacs that store and transport a variety of materials. Inside the nucleus, the DNA is tightly wound around proteins and packaged into compact units called centromeres. All eukaryotic cells are supported by an internal protein scaffold, the cytoskeleton. Although the cells of animals and some protists lack cell walls, the cells of fungi, plants, and many protists have strong cell walls composed of cellulose or chitin fibers embedded in a matrix of other polysaccharides and proteins. Through the rest of this chapter, we'll examine the internal components of the eukaryotic cell in more detail. The largest and most easily seen organelle within the eukaryotic cell is the nucleus, first described by the Scottish botanist Robert Brown in 1831. Nuclei are roughly spherical in shape, and in animal cells they are typically located in the central region of the cell. In some cells, a network of fine cytoplasmic filaments seems to cradle the nucleus in this position. The nucleus contains the genetic information that enables the synthesis of nearly all proteins of a living eukaryotic cell. Most eukaryotic cells possess a single nucleus, although the cells of fungi and some other groups may have several to many nuclei. Mammalian erythrocytes, those are red blood cells, lose their nuclei when they mature. Many nuclei exhibit a dark staining zone called a nucleolus, which is a region where the intensive synthesis of ribosomal RNA is taking place. The surface of the nucleus is bound by two phospholipid bilayer membranes, which together makes up the nuclear envelope. The outer membrane of the nuclear envelope is continuous with the cytoplasm's interior membrane system, called the endoplasmic reticulum. So this shows you all an animal cell. The nucleus is there in the relative center. This shows you a plant cell. And again, the nucleus is there at that relative top center. Scattered over the surface of the nuclear envelope are what appear to be shallow depressions in the electron micrograph, but are in fact structures called nuclear pores. These pores are 50 to 80 nanometers apart at locations where two membrane layers of the nuclear envelope come together. The structure consists of a central framework with eightfold symmetry that is embedded within the nuclear envelope. This is bounded by a cytoplasmic phase with 8 fibers and a nuclear phase with a complex ring that forms a basket beneath the central ring. The pore allows ions and small molecules to diffuse freely between the nucleoplasm and cytoplasm, while controlling the passage of proteins and RNA protein complexes. Transport across the pore is controlled and consists mainly upon the import of proteins that function in the nucleus, and the export of the cytoplasm, RNA, and DNA, protein complexes found in the nucleus. The inner surface of the nuclear envelope is covered with a network of fibers that make up the nuclear lamina. This is composed of intermediate filament fibers called nuclear lamins. This structure gives the nucleus its shape and also is involved in the deconstruction and reconstruction of the nuclear envelope. Chromatin. In both prokaryotes and eukaryotes, DNA is the molecule that stores genetic information, and in eukaryotes, the DNA is divided into multiple linear chromosomes, which are organized with proteins into a complex structure called chromatin. It's becoming clear that the very structure of chromatin affects the function of DNA. Changes in gene expression do not involve changes in DNA sequence. The so-called epigenetic changes involve alterations in chromosome structure, or at least in chromatin structure. Although not still fully understood, this offers exciting new view of old ideas. Chromatin is now more organized than the nucleus than was thought. The state of DNA in chromatin must also change over the course of cell division, as you all will see in Chapter 10. Chromosomes will be compacted into a more highly condensed state that forms the X shape of chromosomes, visible by the light microscope. So you can kind of think of a big bowl of noodles as being that chromatin state of DNA, and then the X that you may have seen in life before being that chromosomal state of DNA. Before cells can synthesize proteins in large quantity, they must first construct a large number of ribosomes to carry out this synthesis. Hundreds of copies of the genes encoding the ribosomal RNAs are clustered together on the chromosome, facilitating ribosome structure. By transcribing RNA molecules from this cluster, the cell rapidly generates large numbers of molecules needed to produce ribosomes. The clusters of ribosomal RNA genes, the RNAs they produce, and the ribosomal proteins all come together within the nucleus during ribosome production. These ribosomal assembly areas are easily visible with the nucleus as one or more dark staining regions called nucleoli. Nucleoli can be seen under the light microscope even when the chromosomes are uncoiled. Although the DNA in the cell's nucleus encodes the amino acid sequence for each protein in the cell, proteins are not assembled there. A simple experiment demonstrates this. If a brief pulse of radioactive amino acid is administered to a cell, the radioactivity shows up associated with the newly made protein in the cytoplasm, not the nucleus. When investigators first carried out these experiments, they found that protein synthesis is associated with large RNA protein complexes called ribosomes outside of the nucleus. Ribosomes are among the most complex molecular assemblies found in cells. Each ribosome is composed of 2 subunits, each of which is composed of a combination of RNA and proteins. These subunits join to form a functional ribosome only when they are actively synthesizing proteins. This complicated process requires 2 other main forms of messenger RNA, or 2 other forms of RNA, one of which is messenger RNA, the other of which is tRNA. So messenger RNA carries the coding information, and tRNA is that carries amino acids. So ribosomes are used in the transfer of mRNA to direct the synthesis of a protein. Ribosomes are found either free in the cytoplasm or associated with internal membranes. Free ribosomes synthesize proteins that are found in the cytoplasm. Nuclear proteins, mitochondrial proteins, and proteins found in other organelles not derived from the endomembrane system. Membrane-associated ribosomes synthesize membrane proteins found in the endomembrane system and proteins destined for export from the cell. Ribosomes can be thought of as universal organelles because they are found in all cell types from all 3 domains. As long as we build a picture of the minimal essential functions for cellular life, ribosomes will be on that short list. Life is protein-based, and ribosomes are the factories that make the proteins. The interior of the eukaryotic cell is packed with membranes that form an elaborate internal or endomembrane system. This endomembrane system fills the cell, dividing it into compartments, channeling the passage of molecules through the interior of the cell. It also provides substances for the synthesis of lipids and some proteins. The endomembrane system in eukaryotic cells is one of the fundamental distinctions between eukaryotes and prokaryotes. The endoplasmic reticulum is the largest internal membrane. The ER is composed of a phospholipid bilayer embedded with proteins. The ER has functional subdivisions described here and forms a variety of structures, from folded sheets to complex tubular networks. The ER may also be connected with the cytoskeleton, which can affect the ER structure, the 2 largest components in the eukaryotic cell. The 2 largest components in the eukaryotic cell are the space inside the ER, called the cisternal space, or the lumen, and the region exterior to it, the cytosol, which is the fluid compartment of the cytoplasm containing dissolved organic molecules and ions. The rough endoplasmic reticulum. The rough endoplasmic reticulum gets its name from the pebbly appearance on its surface. The rough endoplasmic reticulum is not only visible with a light microscope, but it can be seen using the electron microscope. It appears to consist primarily of flattened sacs with surfaces made bumpy by ribosomes. The proteins synthesized on the surface of the rough endoplasmic reticulum are destined to be exported from the cell, sent to lysosomes or vacuoles, or embedded in the plasma membrane. These proteins enter the cisternal space for the first time in the pathway that will sort proteins to their individual destinations. This pathway also involves the vesicles and the Golgi apparatus. The sequence of the protein being synthesized determines the ribosome. So the sequence of the protein synthesized determines whether the ribosome will become associated with the endoplasmic reticulum or remain in the cytoplasmic ribosome. In the endoplasmic reticulum, newly synthesized proteins can be modified by the addition of short-chain carbohydrates to form glycoproteins. Those proteins bound for secretion are separated from the other products and packaged into vesicles that move to the Golgi for further modification and export. The smooth endoplasmic reticulum, with relatively few bound ribosomes, referred to as the smooth ER. The structure of the smooth endoplasmic reticulum ranges from an array of tubules to flattened sacs to higher order tubular arrays. The membranes of the smooth endoplasmic reticulum contain many embedded enzymes involved in the synthesis of a variety of carbohydrates, steroids, and lipids. Steroid hormones are also synthesized in the smooth ER. The majority of membrane lipids are assembled in the smooth endoplasmic reticulum and then sent to whatever parts of the cell that need membrane components. The membrane proteins in the plasma membrane and other cellular membranes are inserted by hormones on the rough endoplasmic reticulum. An important function of the smooth endoplasmic reticulum is to store intracellular calcium ions. This keeps the cytoplasmic level low, allowing for calcium to be used as a lightning molecule. Allowing calcium ions to be used as a signaling molecule. In muscle cells, for example, calcium ions are used to trigger muscle contraction. In other cells, calcium ions released from the smooth endoplasmic reticulum is involved in diverse signaling pathways. The ratio of smooth ER to rough ER is not fixed but depends on a cell's function. In multicellular animals like humans, this ratio can vary greatly. Cells that carry out extensive lipid synthesis, such as those in the testes, intestines, and brain, have abundant smooth ER. Cells that synthesize secreted proteins, for example, antibodies, have much more extensive rough ER. Another role of the smooth endoplasmic reticulum is to modify foreign substances to make them less toxic. In the liver, enzymes in the smooth endoplasmic reticulum carry out this detoxification. In the liver, the smooth ER carries out detoxification of drugs. This can remove substances that have taken in for therapeutic region, for instance, penicillin. Thus, relatively high doses of some drugs are prescribed to offset the body's efforts to remove them. The liver cells have extensive smooth endoplasmic reticulum with enzymes that can process a variety of substances by chemically modifying them. Flattened sacs of membranous sacs called the Golgi body or Golgi complex. These structures are named for Camillo Golgi of the 19th century, an Italian physicist who first identified them. The individual stacks of membrane are called cisternae, and they vary in number within the Golgi body from the last few protists, from one or a few in protists to 20 or more in animal cells, and to several hundred in plant cells. A lot of cisternae. In vertebrates, individual Golgi are linked to form a Golgi ribbon. They are especially abundant in glandular cells, which manufacture and secrete substances. The Golgi apparatus functions in the collection, packaging, and distribution of molecules synthesized at one location and used within another cell or even outside of it. So it sorts those, it packages and distributes. So it sorts, packages, and transports molecules to be synthesized and then transported out of the cell. A Golgi body has a front and back, each with distinct membrane composition. The receiving side is called the cis phase and is usually located near the endoplasmic reticulum. Materials arrive at the cis phase in transport vesicles that bud off the endoplasmic reticulum, and the exit or trans phase is where they are discharged in the secretory vesicles, which you all can see here in figure 4.12. How material transits through the Golgi has been a source of contention. Models include maturation of the individual cisternae from cis to trans, transport between cisternae by vesicles, and direct tubular connections. Although there is probably transport of material by all of these, it now appears that the primary mechanism is cisternal maturation. Proteins and lipids manufactured on the rough and smooth endoplasmic reticulum are transported into the Golgi apparatus and modified as they pass through it. The most common alterations is the addition or modification of short sugar chains, forming glycoproteins and glycolipids. In many instances, enzymes in the Golgi apparatus modifying existing glycoproteins and glycolipids made in the endoplasmic reticulum by cleaving a sugar from a chain or modifying one or more sugars. These are then packaged into vesicles that pinch off from the trans phase of the Golgi. These vehicles then diffuse to other locations in the cell, distributing the newly synthesized molecules to their appropriate destinations. And another function of Golgi's apparatus is the synthesis of cell wall components. So the non-cellulose polysaccharides that form the part of the cell wall are synthesized in Golgi apparatus and sent to the plasma membrane, where they can be added to the cellulose that is assembled on the exterior of the cell. Other polysaccharides secreted by plants are also synthesized in the Golgi apparatus. This has been Golgi. Lysosomes are digestive vesicles that arise from the Golgi apparatus. They contain high levels of a variety of enzymes that degrade proteins, nucleic acids, lipids, and carbohydrates. As these enzymes could destroy the cell, it is critical to segregate them, and then throughout the lives of eukaryotic cells, lysosomal enzymes break down old organelles and recycle their component molecules, allowing room for newly formed organelles. For example, mitochondria are replaced in some tissues every 10 days. The digestive enzymes in the lysosome are optimally active at an acidic pH. Lysosomes are activated by fusing with a food vesicle produced by phagocytosis, or by fusing with an old worn-out organelle. The fusion event activates proton pumps in the lysosomal membrane, lowering the internal pH. As the internal pH falls, the digestive enzymes contained in the lysosome become active. This leads to the degradation of molecules in the food vesicle, or the destruction of the old organelle. A number of human genetic disorders, collectively called lysosomal storage disorders, affect lysosomes. For example, Tay-Sachs disease is caused by the loss of function of a single lysosomal enzyme, hexosaminidase. This enzyme is necessary to break down a membrane glycolipid found in nerve cells. Accumulation of the glycolipid in lysosomes affect nerve cell function, leading to a variety of clinical symptoms such as seizures and muscle rigidity. In addition to breaking down organelles, other structures within cells, lysosomes eliminate other cells that the cell has engulfed by phagocytosis. So when a white blood cell, for example, phagocytizes a passing pathogen, lysosomes fuse with that resulting food vesicle, releasing their enzymes into the vesicle and degrading the material within. And maybe y'all can see that right there before you on that left-hand or right-hand side. These are lysosomes. So very briefly, lipid droplets are organelles with a different structure from the rest of the endomembrane system. They consist of a neutral lipid core surrounded by a single layer of phospholipid. Neutral lipids are hydrophobic and include the triglycerides and steroid lipids. These neutral lipids are made soluble in the aqueous cytosol by being coated with a phospholipid. Peroxisomes. Peroxisomes are microbodies that contain enzymes used to oxidize fatty acids. If these oxidative enzymes were free in the cytoplasm, they could short-circuit cellular metabolism, which involves adding hydrogen atoms to oxygen. So peroxisomes get their name from the hydrogen peroxide produced as a byproduct of the activities of oxidative enzymes. Hydrogen peroxide is dangerous to cells because of its violent chemical reactivity. However, peroxisomes contain an enzyme catalase, which catalyzes the decomposition of hydrogen peroxide into water. Because many peroxisomal proteins are synthesized by cytoplasmic ribosomes, the organelles themselves were long thought to form by the addition of lipids and proteins, leading to growth. As they grow larger, they divide to produce new peroxisomes, although division of peroxisomes still appears to occur. It is now clear that the peroxisomes can still form the fusion of endoplasmic reticulum vesicles, or endoplasmic reticulum derived vesicles. These vesicles then import peroxisomal proteins to form a mature peroxisome. Genetic screens have isolated some 32 genes that encode proteins involved in the biosynthesis and maintenance of peroxisomes. The human genetic diseases called peroxisome biogenesis disorders can be caused by mutations in some of these genes. That is the peroxisome. Plant cells have specialized membrane-bound structures called vacuoles. The most conspicuous example is a large central vacuole seen in most plant cells. In fact, vacuole actually means blank space, referring to its appearance in the light microscope. The membrane surrounding this vacuole is called the tonoplast because it contains channels for water that are used to help the cell maintain its tonicity or osmotic balance. For many years, biologists assumed that there was one type of vacuole with multiple functions. These included water balance and storage of useful molecules such as sugars, ions, and pigments, and waste products. The vacuole was also thought to contain enzymes used to break down molecules and to detoxify foreign substances. So there you have it, the central vacuole. Mitochondria vary in both size and shape, often existing as part of a dynamic network found in all types of eukaryotic cells. Mitochondria are bounded by two membranes, a smooth outer membrane and an inner folded membrane with numerous contiguous layers called cristae. The cristae partition the mitochondria into two compartments, a matrix lying inside the inner membrane and an outer compartment, the intermembrane space, lying between the two mitochondrial membranes. On the surface of the inner membrane and also embedded within it are proteins that carry out oxidative metabolism, the oxygen-requiring process by which energy in macromolecules is used to produce ATP. Mitochondria have their own DNA. This DNA contains several genes that possess proteins essential to the mitochondrion's role in oxidative metabolism. Thus, the mitochondrion, in many respects, acts as a cell within a cell, containing its own genetic information, specifying proteins for its unique functions, and the mitochondria are not fully autonomous. However, because nearly all the genes that encode the enzymes used in oxidative metabolism are located in the cell nucleus. So a eukaryotic cell does not produce brand new mitochondria each time it divides. Instead, the mitochondria themselves provide divide into, doubling in number, and these are partitioned by the new cells. Most of the components required for mitochondrial divisions are encoded by genes in the nucleus and translated into proteins by cytoplasmic ribosome. Mitochondrial replication is therefore impossible without nuclear participation, and mitochondria thus cannot grow in a cell-free culture. Plant cells and cells of other eukaryotic organisms carry out photosynthesis, typically contain from 1 to several hundred chloroplasts. Chloroplasts bestow an obvious advantage on the organism that possesses them. They can manufacture their own food, and chloroplasts contain the photosynthetic pigment chlorophyll that gives most plants their green color. The chloroplast, like the mitochondria, is surrounded by two membranes. However, the chloroplasts are larger and more complex than the mitochondria. In addition, the outer and inner membranes, which lie in close association with each other, chloroplasts have closed compartments of stacked membranes called grana, which lie inside the inner membrane. A chloroplast may contain 100 or more grana. Each granum may contain from a few to several dozen disc-shaped structures called thylakoids. On the surface of the thylakoids are the light-capturing photosynthetic pigments, to be discussed in Chapter 8. Surrounding the thylakoid is a fluid matrix called the stroma. The enzymes used to synthesize glucose during photosynthesis are found in the stroma. Like mitochondria, chloroplasts have DNA, but many of the genes that specify chloroplast components are located in the nucleus. Some of the elements used in photosynthesis, including the specific protein components necessary to accomplish the reaction, are synthesized entirely within the chloroplast. Other DNA-containing organelles in plants, called leucoplasts, which lack pigment and complex internal structure. Root cells in some plants, by way of leucoplasts, may serve as starch storage sites. Symbiosis is a close relationship between organisms of different species that live together. The theory of endosymbiosis proposes that some of today's eukaryotic organelles evolved by symbiosis arising between two cells that were each free-living. One cell, a prokaryote, was engulfed by and became part of another cell, which was a precursor for the modern eukaryote. According to the endosymbiont theory, which I can show the photo now, this is figure 1418, the engulfed prokaryotes provided their hosts with certain advantages associated with their special metabolic abilities. Two key eukaryotic organelles are believed to be the descendants of these endosymbiotic prokaryotes: mitochondria, which are thought to have originated as bacteria capable of carrying out oxidative metabolism, and chloroplasts, which apparently arose from photosynthetic bacteria. This is discussed in detail in the 28th chapter. The cytoskeleton. The cytoplasm of all eukaryotic cells is crisscrossed by a network of protein fibers that supports the shape of the cell and anchors organelles to fixed locations. This network is called the cytoskeleton. It is a dynamic system, constantly assembling and disassembling. Individual fibers consist of polymers of identical protein subunits that attract one another and spontaneously assemble into two long chains. Fibers disassemble in the same way as one subunit after another breaks away from the other end. Three types of fibers compose the cytoskeleton. Eukaryotic cells have those following three types of cytoskeleton fibers, each formed from a different subunit. 1. Actin filaments, sometimes called microfilaments. 2. Microtubules. And 3. Intermediate filaments. So actin filaments, it says two protein chains loosely twined together for movements such as contraction, crawling, or even pinching. So cells regulate the rate of actin polymerization through other proteins as well as switches during polymerization when appropriate. Actin filaments are responsible for cellular movements such as contraction, as I just stated, crawling, pinching, and during division, and formation of cellular extensions. So microtubules, in many cases, they are facilitating cellular movement. So they move materials within the cell itself. And intermediate filaments, they're constituting a mixed group of cytoskeleton fibers. Most common type, composed of protein subunits called vimentin, which provides structural stability for many kinds of cells. Keratin, another class of intermediate filament found in the epithelial cells that line organs and body cavities, and the associated structures such as hair and fingernails. And the intermediate filaments of nerve cells are called neurofilaments. Centrosomes are barrel-shaped organelles found in the cells of animals and most protists. They occur in pairs, usually located at right angles. And you'll learn more about the centrosomes as you do the process known as meiosis or cell mitosis, and meiosis with cell division. So there they are, centrioles by way of the centrosomes. The cytoskeleton also helps move materials within cells. And given that, I believe I'll move on to the next portion on cellular movement by way of cilia and flagella. And this is all your book shows here, so let's begin. So a flagellum is a larger structure. Earlier in this chapter, you saw that bacteria and archaeal flagella evolved independently, and we now see a third kind of flagella evolved in eukaryotes, the eukaryotic flagellum, consisting of a circle of 9 microtubule pairs and 2 central microtubules. So the flagellum you'll find in the sperm cell, allowing the sperm cell to swim. Next up, we'll get to cilia. So other structures called cilia, with an organization of similar to the 9+2 arrangement of the microtubules, can still be found within them. So cilia are short cellular projections, which are often organized in rows. They are more numerous than flagella on the cellular surface, but have the same internal structure. So many organisms' cilia carry out tasks far removed from their original function of propelling cells in water. For instance, beating rows of cilia move water over a tissue's surface. And now we're moving on to cell walls to end the chapter. We're nearing the end of the chapter. The cells of plants, fungi, and many types of protists have cell walls which protect and support the cells. The cell walls of these eukaryotes are chemically and structurally different from the prokaryotic cell walls. In plants and protists, the cell walls are composed of fibers of the polysaccharide cellulose, whereas in fungi, the cell walls are composed of chitin. In plants, primary cell walls are laid down when the cell is still growing, between the cell walls of adjacent sticky substances, called the middle lamella, glues the cells together. Some plant cells produce strong secondary cell walls, which are deposited inside the primary cell walls of fully expanded cells. Animal cells, however, lack cell walls. Instead, animal cells secrete an elaborate mixture of glycoproteins into the space around them, forming the extracellular matrix. So the fibrous protein collagen, the same protein found in cartilage, tendons, cartilage, tendons, and ligaments may be abundant in the extracellular matrix. Strong fibers of collagen and another fibrous protein, elastin, are embedded within the complex web of glycoprotein. So this is table 4.3, and what you have here is a comparison of prokaryotic cells animal cells, and plant cells. You can use this as a quick way to make a comparison between those three and helping with studying. Cell-to-cell interactions. So a basic feature of multicellular animals is the formation of diverse kinds of tissue, such as skin, blood, or muscle, where cells are organized in specific ways. Cells must also be able to communicate with each other and have markers of individual identity. All of these functions, the connections between cells, markers of cellular identity, and cell communication involve membrane proteins and proteins secreted by cells. As organisms develop, the cells acquire their identities by carefully controlling the expression of those genes, turning on a specific set of genes that encodes functions of each cell type. One key set of genes functions to mark the surfaces of cells, identifying them as being of a particular type. When cells make contact, they read each other's cell surface markers and react accordingly. Cells that are part of the same tissue type recognize each other, and they frequently respond by forming connections between their surfaces to better coordinate their functions. So glycolipids, the most tissue-specific cell-to-cell markers are glycolipids, that is, lipids with carbohydrate heads. The glycolipids on the surface of red blood cells are responsible for the ABO blood types, or ABO blood group. Next up, we have the major histocompatibility complex. So it's an example of a cell surface marker that is a recognition of self versus non-self by the immune system. This function is vital in multicellular organisms, which need to defend themselves against invading or malignant cells. So the immune system of vertebrates uses a particular set of markers to determine the self from the non-self by genes encoded in or encoded by major histocompatibility complex. So cell recognition in the immune system will not be covered in this class. So cell-to-cell connections or cell-to-cell adhesion. Adhesive junctions include adherins, and those are molecules on surface of cells. So cadherin superfamily is a large family of calcium-dependent adhesion molecules. So the first cadherin molecules appeared more than 600 million years ago, and with this, cadherin is a single-pass transmembrane protein with extracellular domain that can interact with extracellular domain of cadherin. An adhesive junction. Desmosomes are a cadherin-based junction unique to vertebrates. They contain cadherins, desmocolin, and desmogliin, which interact with the intermediate filaments of cytoskeletons instead of actin. Desmosomes join adjacent cells. These connections support tissues against mechanical stress. Septator-tight junctions. Septate junctions are both found in invertebrates and vertebrates, and they form a barrier that can seal off a sheet of cells. Think in Drosophila, the fly. Tight junctions are unique to vertebrates and contain proteins called claudins because their ability to occlude or block substances from passing between cells. This form of junction between cells acts as a wall between the tissue, keeping molecules on one side or the other. Creating sheets of cells. We won't get too much into creating sheets of cells, but the cells that line an animal's digestive tract are organized in a sheet only one cell layer thick. One surface of the sheet faces the inside of the tract, the other faces the extracellular space where the blood vessels are located. Tight junctions encircle each cell in the sheet like a belt cinches around a person's waist. The junctions between the neighboring cells are so securely attached that there is no space between them for leakage. Hence, nutrients absorbed from food into the digestive tract must pass directly through the cells in the sheet to enter the bloodstream because they cannot pass through the spaces between the cells. This is important to know. Communicating junctions. So gap junctions in animals allow communication. Gap junctions are found in both invertebrates and vertebrates. In invertebrates, they're formed by proteins known as connexins. In vertebrates, however, the connexin-based gap junctions exist, but there is an additional type based on these similar proteins called connections. In each case, the structure is formed by complexes of 6 identical transmembrane proteins. So gap junctions provide passageways large enough to prevent small substances, such as simple sugars and amino acids, to pass from one cell to the next. Yet the passages are small to prevent passage of larger molecules, such as proteins. So gap junctions are dynamic structures that can open or close in response to a variety of factors, including calcium and hydrogen ions. You all need to know this. This gating serves at least one important function. When the cell is damaged, its plasma membrane becomes leaky. Ions in high concentrations outside the cell, such as calcium, flow into the damaged cell and close its gap junction channels, and this isolates the cell and prevents damage from spreading. Plasmodesmata in plants. In plants, cell walls separate from all others. So cell-to-cell junctions only occur at holes or gaps in the walls where plasma membranes of adjacent cells can come in contact with one another. Cytoplasmic connections that form across the touching plasma membranes are called plasmodesmata. And you can see examples of these here: the tight junction, the adhesive junction or desmosome, and then communicating junctions, such as the plasmodesmata, which I am at now. So the majority of living cells within the higher plant are connected to their neighbors by these junctions. So plasmodesmata functions much like gap junctions in animal cells, although their structure is more complex. Unlike gap junctions, plasmodesmata are lined with plasma membrane and contain a central tubule that connects the endoplasmic reticulum of the two cells. So there you have those plasmodesmata. So this has been your instructor.